When we think about the blueprint of life, our minds almost always jump to the massive genome that's packed away inside the nucleus of our cells. But that isn't the whole story. Today, we're going to take a closer look at another absolutely critical piece of our genetic architecture, human mitochondrial DNA.
So that leads us to a fundamental question. Is the nuclear genome really the only one that defines us? The answer, perhaps surprisingly, is a firm no. See, there's another genome, a second one, hiding in plain sight within the power centers of our cells, and it's incredibly important.
And this second genome is, of course, mitochondrial DNA, or mtD, as we'll call it. Its home isn't in the nucleus with the rest of our chromosomes. Instead, it's found inside the mitochondria themselves, the organelles that you probably remember as the powerhouses of the cell.
Now, what's so fascinating here is its primary job. While the nuclear genome is busy conducting the grand orchestra of the cell's identity and development, mtDNA has an almost singular focus on one task: generating ATP, adenosine triphosphate. And that, right there, is the essential energy currency that fuels basically every single thing our bodies do.
To really appreciate its role, we first have to understand its unique structure, because, as you'll see, it's profoundly different from the DNA we're all used to hearing about. Let's start with a number: 16,569. That's roughly the number of base pairs in the human mitochondrial genome. It's incredibly compact. And to put that into perspective, our nuclear DNA contains over 3 billion base pairs. It's a staggering difference in scale. And this table just lays out those differences so clearly. Nuclear DNA is linear, like threads, and kept in the nucleus. But mtDNA is a small, circular molecule, just hanging out inside the mitochondria. It has only 37 genes, compared to the 20,000 or so in the nucleus. But perhaps the most remarkable distinction of all is its inheritance pattern. mtDNA comes exclusively from your mother.
So what do these 37 specialized genes actually do? Well, 13 of them provide the code for key proteins in the electron transport chain, which is the core engine of cellular respiration. The other 24 genes create the RNA machinery, the tRNAs and RNAs that's needed to actually build those very proteins. It's a beautifully efficient and self-contained system dedicated entirely to energy production.
And this brings us to one of the most fascinating aspects of mtDNA, that unique way it's passed down. Let's really dig into the mechanics of this strictly maternal inheritance, because its implications are huge. Okay, so here's what happens. During fertilization, the sperm delivers its nuclear DNA to the egg, but any mitochondria that hitch a ride with the sperm are essentially tagged for destruction and eliminated very quickly. The end result is that the developing embryo gets its entire population of mitochondria and all of its mtDNA from the mother's egg cell and the mother's egg cell alone. Now, because this DNA is passed down from mother to child without being shuffled or recombined with paternal DNA, it acts as a nearly perfect historical marker. This property has made it an incredibly powerful tool for population genetics. It lets scientists trace human migration patterns across continents and even helped us identify a common maternal ancestor for every human alive today, a concept you might know as mitochondrial Eve.
But it's not all straightforward. The mitochondrial genome has its own vulnerabilities. Its unique structure and location make it especially prone to mutation, and this can lead to some very complex biological situations.
You see, our nuclear DNA is tightly wrapped around protective proteins called histones. Think of it like precious cargo being carefully packaged. Mitochondrial DNA, on the other hand, lacks this protection. It's essentially naked. That structural vulnerability, plus its close proximity to the high-energy, free radical-producing environment of the respiratory chain, means it has a much higher mutation rate than nuclear DNA.
These frequent mutations can lead to a state known as heteroplasmy. This is where a single cell can contain a mixture of different mtDNA versions, some normal or wild type, and some mutated. And it turns out that the ratio, the specific percentage of mutated versus normal mtDNA, can be a really critical factor in the research of many complex disorders tied to mitochondrial function.
So you can see how this presents a massive challenge for researchers. If you want to understand the role mtDNA plays in our biology, you have to be able to detect these tiny variations with extreme precision. So the question becomes, how can you possibly quantify that exact ratio of mutated to wild-type mtDNA with any accuracy?
Well, the answer, as is often the case in modern biology, is found in advanced technology. To really tackle the unique challenges of analyzing mtDNA, you need specialized, high-throughput sequencing solutions.
This is where specialized services, like the human mitochondrial DNA sequencing offered by CD Genomics, have been developed to meet this precise need. They use targeted approaches like multiplex PCR or probe capture to zero in on and enrich the mitochondrial DNA, allowing for an incredibly comprehensive and ultra-deep analysis.
This kind of methodology brings some major advantages to the table. For one, researchers can use total genomic DNA, which means they get to skip the very difficult step of trying to purify mtDNA from cells. It also achieves what's called ultra-deep sequencing. We're talking greater than 1000x coverage. That's absolutely essential for accurately detecting variants that might be present at a very low frequency. Plus, it ensures 100% coverage of the entire mitochondrial genome with high accuracy, all with a pretty fast turnaround.
And if you look at the technical specs, you can see these services are designed for really robust and reliable performance. They often use a workhorse platform like the Illumina Chieve 150. They only require manageable amounts of input DNA, and they achieve a high capture efficiency. That just ensures the vast majority of the sequencing data you get back is actually from the mitochondrial genome you're trying to study.
But, of course, getting the raw data is only half the battle. The real value often lies in the analysis that follows. A standard bioinformatics pipeline for this kind of work covers everything from quality control and alignment all the way to calling out and annotating genetic variants. And most importantly for this field, it can also offer those specialized analyses for heteroplasmy levels and for determining haplogroups, giving a researcher a truly complete picture.
And this all brings us to one final thought. As the tools we have to probe this unique little genome become more and more powerful, more and more precise, it really makes you wonder, what fundamental questions about human evolution, about metabolic function, about the very origins of complex disorders will the deep analysis of our mitochondrial DNA finally allow us to answer?